All right. Hope everybody is well rested after fall break.
Oh, yeah.
Yeah?
So much.
Much needed.
It's at least nice to have a few days.
I slept 13 hours.
Okay. So I want to sort of go through what the next half of our semester looks like. So exam 2 sort of marks the end of one of the major units in this class. If you remember the first lecture, which we didn't really go through together, but there are 3 sort of major components in the field of genetics. So there's transmission genetics, there's molecular genetics, and then there's population genetics. We spent half of the semester in transmission genetics, half of the semester in molecular genetics, and then basically 1 week to cover all of population genetics. Which is sad because that's the thing I love the most. But we do what we have to do. So we are now sort of done with the major transmission part, which is Mendel and all that fun stuff, and we're moving into molecular genetics. So we're going to spend the next 6-ish weeks talking about molecular genetics, which most of you are probably already somewhat familiar with. That's our DNA structure, replication, transcription, and translation. I clearly did not rest enough over fall break. So we are switching to kind of a different way of thinking. Some people find this more intuitive than transmission genetics. Some people don't, so we'll do our best to sort of stay together. We are also switching over in lab, so we're switching away from sort of our, again, Mendelian sort of recitation style lab and moving into molecular techniques. Some people, who's had cell molecular already? Okay. So for most people, this will be an introduction to molecular techniques that then, if you take cell molecular, continue.
So we're doing like PCI and stuff?
We are. Yeah, so this week in lab, if you saw posted, you're going to basically be running through sort of the experimental design piece as well as defining and trying to understand the techniques that we're going to be doing over the next 6 weeks. And then you're going to be practicing pipetting, which, yay, moving liquid from place to place. But that's a skill that we're going to make sure that we're going to need to make sure that we know really well before we move into next week's lab, which is DNA extraction. So I'll remind you that starting next week, we are going to be dealing with like actual reagents and chemicals and stuff. So I'll tell you sort of week by week what the safety precautions need to be or what you need to bring to lab or how you need to be prepared. So we'll go over that in lab this week. I think that's all I have.
Should we not be in session?
No, tomorrow you're just going to be reading and then doing pipetting practice. It's going to be the last, like, not super exciting lab to do. Nice. Okay, all right, so I think that's all I have to sort of catch up on. I did want to talk a little bit about, or sort of get a feel for what your all's finals weeks look like. So I know that this year is the first year we're moving to a last week of class as our finals week model. Is everybody aware of that? Okay, so we don't have specific finals time slots. I currently am envisioning basically small exams. So we have a cumulative exam portion and then we have a new materials exam portion that I usually put in one as the final. So I was going to divide it up over days and then cancel lab that week and have lab as either you can come and talk to me about your final lab report or I can run like review sessions on those days for whatever exam part's coming up the next day. The labs would be open to everyone on both days, so you wouldn't just come on your lab day, but you could come either Tuesday or Thursday depending on what you want to review, since Wednesday would be one exam and Friday would be a different material set on an exam. But I didn't know what people's current exam week looks like.
I have no idea. Yeah, no
This is the
idea.
first we talked about it in the class.
Yeah.
Oh, okay.
Yeah.
Well, then if I'm first, then I get to decide what we do, right? Okay, so I'm going to leave it as that, and then essentially what I'm saying is I'm not going to ask for an exam block outside of class time, which some of your instructors may do. You may have an actual finals exam block for a class. I'm not going to do that. I'm just going to somehow spread out that final exam over a couple of class periods. And then as we get closer, I'll figure it out a little bit more specifically. But that last week of class, basically, we're not covering new content. We'll be assessing our learning. Okay, and there's only one more exam between now and then. So, I don't know if that's terrifying or not. All right. Okay, is there anything that I'm missing? No, we're good to go? All right. Okay, so since we are switching from transmission to molecular genetics, I don't remember what this link is for, so let's find out. Could be exciting. Definitely genetics related for sure. It is. Yeah, so this I think is like the coolest video. It is sort of a compilation of animations for what these different molecular, all the molecular machinery within our cells looks like. So we don't often sort of visualize it this way, but I think it's really very cool. Yeah, there's a lot of cell and a lot of sort of stuff in here.
These are tiny molecular machines, and they are doing this inside your body right now. Do you understand why we have to zoom out? Every day in an adult human body, 50 to 70 billion of your cells die. Either they're stressed or damaged or just old. But this is normal. In fact, it's called programmed cell death. But to make up for all these lost cells, right now, billions of your cells are divided, essentially creating new cells. And that process of cell division, also called mitosis, well, it requires an army of tiny molecular machines.
Yeah?
So let's take a closer look. DNA is a good place to start, the double helix molecule we always talk about. This is a scientifically accurate depiction of DNA created by Drew Barry at the Walter and Eliza Hall Institute of Medical Research. If you unwind the two strands, you can see that each has a sugar phosphate backbone connected to the sequence of nucleic acid base pairs, known by the letters A, T, G, and C. Now the strands run in opposite directions, which is important when you go to copy DNA. Copying DNA is one of the first steps in cell division. Here, the two strands of DNA are being unwound and separated by the tiny blue molecular machine called helicase. Helicase literally spins as fast as a jet engine. The strand of DNA on the right has its complementary strand assembled continuously, but the other strand is more complicated because it runs in the opposite direction. So it must be looped out with its complementary strand assembled in reverse, section by section. At the end of this process, you have two identical DNA molecules, each one a few centimeters long, but just a couple nanometers wide. So to prevent the DNA from becoming a tangled mess, it is wrapped around proteins called histones, forming a nucleosome. These nucleosomes are bundled together into a fiber known as chromatin, which is further looped and coiled to form a chromosome, one of the largest molecular structures in your body. You can actually see chromosomes under a microscope in dividing cells. Only then do they take on their characteristic shape. Otherwise, the DNA is more strewn inside the nucleus. The process of dividing a cell takes around an hour in mammals, so this footage is from a time lapse. You can see how the chromosomes line up on the equator of the cell. Now, when everything is right, they are pulled apart into the two new daughter cells, each one containing an identical copy of DNA. Now, as simple as this looks, the process is incredibly complicated and requires even more fascinating molecular machines to accomplish it. So let's look at a single chromosome. One chromosome consists of two sausage-shaped chromatids, containing the identical copies of DNA made earlier. Each chromatid is attached to microtubule fibers, which guide and help align them in the correct position. The microtubules are connected to the chromatid at the kinetochore, here colored red. The kinetochore consists of hundreds of different proteins working together to achieve multiple objectives. In fact, it's one of the most sophisticated molecular mechanisms inside your body. The kinetochore is central to the successful separation of the chromatids. It creates a dynamic connection between the chromosome and the microtubules. For a reason no one's yet been able to figure out, the microtubules are constantly being built at one end and deconstructed at the other. While the chromosome is still getting ready, the kinetochore sends out a chemical stop signal to the rest of the cell, shown here by the red molecules, basically saying this chromosome is not yet ready to divide. The kinetochore also mechanically senses tension. When the tension is just right and the position and attachment are correct, all the proteins get ready, shown here by turning green. At this point, the stop signal broadcasting system is not switched off. Instead, it is literally carried away from the kinetochore down the microtubules by a dynein motor. That's the walking guy. This is really what it looks like. It has long legs so it can avoid obstacles and step over the kinesins, molecular motors that walk in the opposite direction. Personally, I'm astounded by these tiny molecular machines, how they're able to routinely and faithfully execute their functions billions of times over inside your body at this exact instant. I'm also amazed by the scientists who were able to...
Okay. So, we're definitely starting to look at things at a different level, right? This is a little bit different from transmission genetics. It's actually sort of getting smaller, right? We're looking at the smaller sort of bits and pieces now. But I think it's really often that we sort of think of these things as being static, and really they're not static. They're very involved and kind of constantly moving and very cool. Okay, so we are going to start by looking at a couple of experiments that were done sort of historically that actually made us aware that DNA is our genetic material. So keep in mind that in the 1800s, we didn't know about chromosomes and we didn't yet know about DNA. We certainly didn't know what the inheritable unit was. So in Mendel's time, we just called them basically units of inheritance. At that point, we were unsure what they were. So I'm going to have us divide them into groups here in a few minutes and do some not listening to me talk activities. But first, I just wanted to list the criteria for something to be a genetic material. The first thing is that it has to contain information. Right? And that information is going to be all of the instructions that are necessary in order to make a living organism. So we always talk about DNA as sort of being the blueprint of the body. The information that's contained in the DNA is what gives an organism its physical characteristics. I just watched a movie on Sunday recently. Has anybody seen Annihilation? It's this weird movie where they're in this mist-like stuff, but apparently it refracts all this information back into itself, and so there were plants that were growing in the shapes of humans because apparently they had Hox genes now in the plants, because all the DNA from humans got like reverberated back into plants. It was super weird. Anyways, when I was talking about body plans and organization, I thought about these plants growing like humans. Yeah, so it has to have information. It also has to be transmissible, right? So it has to be able to be passed from a parent to an offspring. We've already talked about that at length. It must be capable of replication. We touched on that when we talked about mitosis and meiosis, right? That first sort of interphase piece is going to be cell growth and then DNA replication. If we don't replicate it, then we can't pass it to the next generation without removing it from ourselves, which doesn't work if it is the information that makes us what we are. And then the last piece is that there has to be variation. So we have to be able to change it. There has to be variation within a population or within a species as well. So genetic variation is the foundation for what process?
Natural selection.
Evolution by natural selection. Excellent. Yes. Very good. Okay. Okay, so what I want to do is have us break up into 3 groups, and we're actually going to move the furniture in this room as was intended when this room was designed, which I haven't done in here yet, so I'm excited. So you're going to move tables, you're going to make groups of 7 or 8, so I see sort of maybe a natural group in the front, and then this middle row plus a person, and then the back corner as a group. So turn desks, or if you want to like turn and face the people behind you, that would work too. Divide yourself into groups of 7 or 8. I am indifferent to who is with you. Okay. Alright, so this is a group. You guys want to join? Can we get one more in this pod? And then will you two join this grouping? Okay, so what we're going to do is I have 3 experiments that are sort of the foundational 3 pieces to discovering what is the genetic material. So we knew that there were proteins in a cell, we knew that there was DNA in a cell, and we knew that there was, what am I missing? Well, proteins and DNA were RNA. We knew that there were all these things in a cell, but we didn't yet know which one was actually our genetic material, right? And so these are 3 scientists or groups of scientists who were involved in figuring out what our genetic material was. So each group is going to take one of these and lay out the basic information for us. So I brought some extra textbooks. You can also use computers or phones, but you'll want to say sort of what their hypothesis was, how they tested it, so the most important piece is going to be the experimental piece, and then what their results and conclusion were. So we sort of want to walk through the scientific method of what they did. Make sure everybody in your group knows and can explain those pieces, because in 10 to 15 minutes we're going to reform our groups with representatives of each of the 3. And then your job will be to explain your group's experiment to the other members. So by the end, everybody should be in a group with somebody that can explain all 3 of these and sort of understand the foundational experiments that allow us to determine DNA is our genetic material. Okay, so does anybody have a preference of Frederick Griffith? Anybody feel strongly towards any of these people? Avery McCarty McLeod? All right, 1, Frederick Griffith.
I already started.
Avery McCarty McLeod. And in the back, Hershey Chase.
So we're just looking up like everything about this.
Yeah, so you want to look up what their question was,
Bacteriologist.
what their experiment was, and what their results and interpretation were.
Wow.
Pause this.